Okay, it's Monday. This is Thinktech. We're on the SOEST Hour, and the SOEST Hour is sort of like this large umbrella covers so much more at the university. And we have with us in the gallery back there, we have Talia Ogliori, who comes and helps us on behalf of SOEST and UH-Manoa. Thank you, Talia. And she's waving back. Let the record reflect she's waving. Okay, we're talking about research revelations at UH-Manoa with two award-winning researchers, if you will. One a master's candidate and one a PhD candidate. Our PhD candidate to my left is Jabran Zahid, who is with the Institute for Astronomy. Hi, Jabran, say hi.
Hello, hello.
I knew you'd say that. Okay. And our master's candidate is Chino Cabalteja.
Yes, sir.
My handwriting is the only problem. Chino Cabalteja, and he's with CITAR, the College of Tropical Agriculture and Human Resources.
Yes, sir.
Both of these gentlemen are researchers, and we're going to talk today about research at UH-Manoa and what it takes to be excellent. So let's set the stage, gentlemen. And can you tell the people your background, how you got to be where you are today? And when did it dawn on you that you wanted to be in your field, that you wanted to do research, that you wanted to study the things you're studying now? Okay, you go first, Jabran.
All right, well, first of all, thank you very much for having me on the show. It's a pleasure to be here, to be able to discuss my research. I got started in the path that I'm on now about halfway through undergrad. I was pretty good at math when I was in high school, and when I got to college, I studied physics and mathematics. And about halfway through, I realized the part of physics that I really liked was astrophysics.
Where'd you go to college?
University of California, Berkeley. And so then I started focusing on astrophysics, and I did research at the same time. And that's when I really became excited about the prospects of doing research level, pursuing a degree that allowed me to do research. And so I took a little break after college, and then came here to the University of Hawaii to study astrophysics.
Well, you know, IFA has got some people with high-minded people.
Yeah.
I say that.
I think that's a compliment.
They all have a kind of apotheosis kind of story about how they decided to study the heavens. And we did a movie recently about IFA. The open house, Astronomy open house, about 3 weeks ago.
Yeah, great.
And it was a great movie because the kids and the astronomers and the friends of the astronomer, friends of IFA, it was a fabulous coming together. And the building is a beautiful building. It's perfect for that kind of community affair. But we talked to a lot of them, the researchers and the faculty in general about what made them go to astronomy in the first place. And they all had different and very interesting answers. So what was it, that moment, where you had your experience, your apotheosis?
Yeah, I mean, certainly I think just looking at the stars is an inspiration in its own right, and realizing that I had the tools and the skills and the opportunity to study them. So I always, as a kid, looked at the sky, and I think everyone did. I lived in a desert, so it was quite dark. I could actually see the sky. And I think, seeing that and just wondering what was going on. And so I think, once I had the opportunity and I took a few courses and I realized, wow, this is actually really cool stuff, it was pretty much, I fell in love. And from there on out, it was, I would say the thing I enjoy most about research, which a lot of people don't actually appreciate, is that it's a very creative thing. There's, a lot of creativity involved, and in that sense, it's really, to be able to bring creativity to the study of something so beautiful, I feel like very fortunate to be able to.
Just like the Thomas Edison thing, where you have to go through different elements until you hit the right one, and you have to use your creativity to check all the possible combinations of elements, and then one day, aha.
Well, no,
or is it something else?
I think it's a little different than that. I think it's because of the fact that when we study science, we see it in a textbook. It all seems so logical. So we think when you're doing science, you're just sitting there doing A, then B, then C, then D, because it all seems so logical. But when in fact you're doing science, it doesn't work that way. It's only in retrospect that you could put all the pieces together and present it as this clean, really clean picture of how the world works. So when you're actually doing it, you have to be creative because you're pushing the envelope. You're doing stuff that hasn't been done before, and who knows what the right thing to do is. So the creativity comes in when you want to ask, okay, I have this question I want to ask. How do I go about answering it? And that requires really something new because you're pushing the edge of what we currently know.
Can you take a minute and just give me an example of a creative thought that pleased you?
So, I mean, just most recently, I just put out a paper, and the thing was that there was this one critical step that I needed, which was to do a particular to derive a particular mathematical model. And the key was realizing that, there were certain aspects of this model that had to be true in order for it to be useful to me. And that step was something that hadn't occurred to anyone before. And having done that, I then later, when I applied that model, found that it helped to explain so much more than what the model itself was by itself. And so I think the aha moments happen when you do something in science and it turns out that it ends up explaining so much more than what went into it. And that's, I think, a very important aspect of science, when you can predict things based on the work that you're doing. And so this is just something that happened recently to me. It didn't happen in an instant. It was over several months of very rigorous work. But once you step back and you see it, and it looks very simple, it's only like 5 or 6 equations, but it took me 2 or 3 months of working on it to come up with this. So I think, that's an example of this sort of, moment when you realize, oh, this is the right thing. And of course, it has to be this way.
Yeah, yeah. And you're not passionate about any of this?
No, not at all. I could take it earlier. No, of course, I love what I do. So.
Well, let's talk to you, Chino. So you're in SITAR, and IFA is not specifically in SOEST, and SITAR, the College of Tropical Agriculture and Human Resources, not in SOEST either, but you guys have an affinity, sort of the scientific community at UH-Manoa, I guess I would say. So he looks at the stars, and you look at the small chemical reactions and microbes and toxins and whatnot. So how did you get involved in what you do?
Oh, yes, sir. Thank you very much for having me. Well, my interest in science was fostered at a very young age. I grew up in Kauai, and Kauai has a rich diversity.
They have agriculture.
Of flora and fauna, and I remember, going through the cane fields and chasing some chickens and wondering why can't chickens fly, and that got me very interested in.
Did you ever find out?
Fortunately, my path diverged. And, yeah.
They're still not flying. Yeah.
Yes, and in college I went to UH and majored in microbiology and started to get into the nitty-gritty sciences. After a while, I worked as a phlebotomist.
Before you get to that, what is actually microbiology? What is that?
Microbiology is a study of microbes such as bacteria, viruses, fungi.
So it's not small biology. It's not like microchip or anything like that.
No.
It's micro, like in
microbial organisms.
Microbial organisms.
Yes, sir.
Buggy things.
Yes, sir.
Okay. And it's the biology of those little buggy things.
Yes.
Okay. Just wanted to be clear about that.
Yeah, so I started working a couple of years a year as a phlebotomist.
A phlebotomist?
Yes, sir.
Phlebotomist. I'm afraid that's not in my vocabulary.
Oh,
what is that?
I used to draw blood in the hospitals and send them out for medical testing. And I saw a lot of the patients that came through our clinic, and started to wonder about the biochemical aspects of disease. And when I started my master's program, I was fortunate enough to have gone to the lab of Dr. John Paul Bingham, who does work on bioactive peptides.
A moment on John Paul Bingham. He was here. He's been here a couple times at this table. In fact, we made a movie about him in SITAR. Yeah. With the snails. Scary movie. It's a movie you don't want to play on Halloween. Put your hand in that tank. Whoa.
Oh, yeah.
Snails have all kinds of antigens in them, don't they?
Yeah, they have a venom. We studied the venom of cone snails, and
that's the worst one, isn't it?
Yeah.
That's the meanest.
Yeah. Well, there's several cone snails found throughout the Pacific, and several that are found here in Hawaii. Yeah. And what they found out maybe 30 years ago was that when you look at these the composition of these venoms, you find that they're comprised of peptides, and these
Qu'est-ce que c'est peptide?
Peptides are basically amino acid chains. So amino acids are strung together in a chain, just like like pearls on a necklace. And what happens is they start to fold upon themselves, and each amino acid starts to interact with each other. And these three-dimensional conformations are just the right fit for a bunch of receptors that we have in our body. And when they hit them, such as when the venom hits the fish that they're eating, it causes paralysis
Yeah.
and even death.
That was a guess. Yeah, that's pretty exciting stuff.
Yes, sir.
So then, now peptides has nothing to do with Pepsi-Cola or anything.
No, sir.
And tides has nothing to do with a household soap.
No.
What's the derivation of the term? Or is it just, it's unique, it's just a term in biology?
Well, peptides are basically, they're small chain proteins.
Okay.
So about 10 to 50 amino acids in length. And then if the amino acid chain gets even longer, we call them proteins.
How can you look at it?
We look at it through their profile in a machine called the HPLC, high performance liquid chromatography. And what we do is we inject it, and it absorbs or sticks to a stationary phase. And what we do is we run liquids through it, a liquid phase through it, and see when the molecule comes out. And because of this machine, we're able to analyze the composition of these venoms.
Okay, and because of these machines, we're able to take a break. We're going to take a short break. That's Chino Cabalteja. Cabalteja, it's my handwriting. And Jabran Zahid, okay, respectively from Sitar and IFA. Here in the SOEST Hour on Thinktech, we're talking about research revelations at UH-Manoa. We'll be right back.
Hello, I'm Martin Despang, and I'm the host together with the one and only Ali Amashta, and our show is called Urban Transcendence. And Urban Transcendence is about identifying where we have a unique situation of a vibrant city in one of the most beautiful natural environments. So how these two can coincide, sometimes conflict, how they could find reciprocity in the 21st century is what we're excited about. And we're planning on bringing in a diverse body of guests, both from the arts and the science and the established and the wise and the emerging generation. So hope you will join us. We'll always be on Thursdays from 1 to 2 p.m.
It's Monday, therefore it must be the SOEST Hour here on Thinktech. We're talking about research revelations at UH-Manoa with Jabran Zahid of the Institute for Astronomy and Chino Cabalteja,
Yes.
okay, of Sitar. So we were talking with Chino about why he got into what he got into. I mean, what is it about this with the little buggies and the peptides and the venoms that attract you? Is it a sense of wanting to be close to danger?
In our lab, we're motivated by finding bioactive peptides that can either be used as pharmaceutical leads or pesticide leads. These conotoxins come from venomous cone snails, and these cone snails feed on different things. Some feed on fishes, some feed on worms, and some feed on fellow mollusks. And when we milk them, we get to see what receptors they interact with. And the ones that feed on fishes, the fishes share similar receptors to humans. So we mine these for potential drug leads.
Milk. Sounds like such an interesting verb here. How I saw this happen, but I'll ask you, how do you milk a snail?
Personally, like I'm scared to touch touch the snails, but I've seen them do it a couple of times, and they take an Eppendorf tube, basically a plastic test tube,
yeah.
and they cover it with latex. Yeah. And what they do is they dangle like their food in, usually a goldfish, and they have it inject through the thinnest part of the goldfish, and
So the silly snail doesn't realize he's injecting the tube, not the fish.
yes.
And then you fool him.
Yes.
I saw that happen in John Paul's laboratory, which is a laboratory where you must work. Huh?
Yes.
It's quite a large place.
Yes.
It's filled with fish tanks.
Yes.
Well, so you guys have something in common, and that is you both won a campus-wide competition for excellence in research. Can you tell me how that worked? I mean, did you apply, or did they come to you one day and say, Chino, you're really a special guy. We want to give you this award. How did that work?
From my understanding, we had to be nominated by our advisor. I believe Dr. Bingham nominated me, and we needed to get a couple more letters of recommendation, and we also needed to turn in a comprehensive abstract of our work. Yeah.
So, but what's the test? I mean, we had a funny discussion before the program began. It was excellence, and I said, exactly. Can you help me with what the excellence means? So you're an excellent researcher. That's what they said.
I don't think I'm excellent. But,
Somebody else does that. What is an excellent researcher? I mean, how do you denote an excellence in research? Is it thought process? Is it laboratory technique? Is it choice of subject? Is it the ability to write all this down? Is it all of those things? What is it?
I think it's the ability to face the challenges without losing motivation, because as Jabran said, there's a lot of challenges in science, and it all matters in how you look at it and how you tackle the questions.
So how big a deal is it that you got this award?
I don't know. Do you want to answer that?
Are you proud? Did you tell your family to have a party?
I'm pretty happy to have received it.
Okay.
It's a very humbling experience.
I have a feeling it got you on the show. Only Talia knows for sure. Okay, Jabran, what about you? I mean, do you have the same view of the matter? I mean, what in your mind, what is an excellent researcher?
Well, I think, as I was saying before, I think creativity is really what distinguishes the best researchers, and that's something I think that I don't know where it comes from, inspiration, where that comes from. So that part of it's really important, and I think I've been very fortunate. I think I've had people who support my work that consider it to be very creative, and I think that largely contributed to me winning the award.
So you have to build a network of people who do support your work, right?
That's
It's relations.
exceptionally important.
You know, they
in
say
science.
real estate is not, in the law profession, real estate is not about land, it's about relationships.
Yeah.
As a matter of fact, so is everything about relationships. Even research is all about relationships.
It's not what you know, it's who you know, right?
Yeah.
But no, there is the side about what you know, and that really comes out in the papers that you publish, the studies that you publish in journals that are peer-reviewed. That aspect of research is probably the most important metric of success these days in the way that science has been going. So, being able to publish results and get those results noticed is absolutely crucial within the scientific community, especially for young researchers who are trying to establish themselves in an environment that's very competitive. And since research has grown over the last 20, 30, 40 years, you're going to encounter committees that are either hiring you or giving you awards or whatever that don't know you personally. So then they will rely on your letters of recommendation, but then they'll also just look straight at the numbers. How many papers have you published, and things like that.
Publish or perish, yeah.
Exactly, and that holds very strongly, I would say, in my experience, for example, being on the job market, who gets jobs, things like that. So I think that's another aspect of excellence in research. So
Who's on the committee? I mean, that found both of you guys. Is it the same committee, different committee? Is it campus-wide that found you guys excellent researchers?
So the award was given by the University Research Council. I am not sure who's on the committee. I believe it's
Vasilis Simos?
I'm not actually sure. I don't know the committee, but I know from the IFA, the Institute for Astronomy side, there's first a search within the institute, and then they put forward one candidate. I don't know if that's how it works in your department as well.
I'm not quite sure how it works for CTAR.
Yeah.
Okay.
So it's
The other thing I wanted to ask that came out of this discussion here just now is when you publish, the fact that you publish is no different than what you would be publishing if you were already a PhD, if you're already a notable research faculty in any school in the world. You're going out into the nether with your papers for the entire scientific world to see. There's nothing private about it.
No.
Yeah.
And once it's there for good. So your name's on there, you got to make sure you will really stand behind it. And I think at our young age, that's a lot where the advising comes in, where your advisor will make sure that, you know, you're not, you're sort of taking the care that's required, and there's much more care than I think most people realize in publishing a paper, you know.
Tell us about it. I mean, so one day you wake up and you have a yellow pad or a dictaphone or something. I do dictaphone myself.
Okay.
And you put all those words down, and it's, well, your thoughts are on the paper. Now, with all of that raw material, how do you take that to a paper that's publishable? What do you got to do?
So for me, the process has actually evolved substantially as I've gone through my PhD. The way I do it now is the most important thing in my field is the figures that you show in your paper. The figures should tell the story without any requirement of text, just explanation. So what I do now is
tabular.
It's just figures. It's the plots on of data and things like
charts
that.
And graphs.
Charts and graphs mainly. So I basically create those first. Once I have the results, I create those and display them in figures. I put the figures into a document, and then I write the story around that. In the past, I didn't do that, but I find that you have a much more cohesive and coherent end result and paper when you start from the beginning and just write the story as it's told by the figures.
And sure, and because the guy who reads it, he's going to be looking at the charts and graphs the way it always works. And that's where that's the first thing he looks at.
Exactly.
So he'll look at your text second.
Yeah, if he looks at it at all.
Right. That's the way it is in science now, isn't it? It's all numbers. You know?
I mean, in my field, 20 papers, 20, 30 papers are coming out a day. I don't have time to read that many papers, and the reality is
worldwide.
Worldwide, yeah. And it's now with the internet, this community is so connected that the day after it's submitted or accepted to the journal, it's on the internet for anyone to grab, which is a beautiful thing, and it's really moved science in a very new direction, but it's also created a sort of information overload to some degree. And so exactly what you said, when I look at a paper, the first thing I'm going to do is read the title, and then I'll read the abstract if the title's interesting.
They have abstracts.
Yes.
This is really important. In the law reporters and law encyclopedias, they always have an abstract at the top.
Yeah.
Because if you had to read every case fresh, you'd be busy.
Yeah.
I mean, forever.
Yeah.
So the abstract really helps you. Do you write the abstract, or does somebody else write the abstract?
All my papers that I'm the first author on, I write the whole thing.
What is it to be first author?
So typically you put the author order. I think it's different in different fields. In biology, I'm not sure, but in my field, you put the author order and basically the amount of work that each particular author contributed to that result or that paper.
So the first author would write the abstract?
The first author would be the person who probably came up with most of the ideas, did most of the work, and actually wrote most of the paper. Sometimes people share it, but my experience has been as a PhD student, I typically just like to have control, so I write it.
And the abstract goes to meta tags. So if I'm searching on some sort of scientific search site, then it'll look for those meta tags. No?
Yeah.
Now, does it look full text as well?
It can. I believe Google Scholar, which is going to be the next big thing, does actually go parse through the text.
Why do you say next big thing? It doesn't exist now.
It's there now, but it hasn't quite. I think it's not. I think each of the scientific communities, I can't speak for biology. Maybe you can tell us more about that, Chino. But in my field, there's NASA runs a repository for all the papers, and so they're all there. And then there's an archive, which has been revolutionary. And I think biology has this as well, where anyone, anywhere in the world can post to the archive, and that is anyone can access it too. There's no paywalls behind that journals may put up. And Google is now.
Can I go on there right now and look at Google Scholar?
Yes, you could, and you would find all of my papers there.
I might do that.
Yeah, you're welcome to.
One last point, and I'll move back to Chino, is that in the law, when we write anything, whether it be a brief for court or an article, whatever, we have to follow certain citation conventions, and you can't just describe a periodical without giving its official name and a bunch of information about it, or a case or any authority at all. Is that the same thing?
Yeah, so I think the point of the citations is if you're going to make any claim that's not just obvious fact, that everyone knows, you need to cite where that claim is coming from. And so typically a paper for me will have 50 to 100 citations of various aspects of the work that I didn't myself do, but my work depends on. And since science is an enterprise that builds on previous results and previous work, it's very important to appropriately cite the, where those results are coming
Oh,
from.
this sounds just like the law.
It must be very
It
simple.
also sounds like, and this goes to your dissertation defense, it also sounds like you have to advocate for your position. You must create your position, you must verify your position, you must articulate your position, and then you have to advocate for it so people believe you.
Yeah, so I go to conferences, several conferences a year, presenting my work, arguing for my results, and then arguing with people who don't believe my results, and arguing with things like that. And that's scientific discourse at the heart of it. Is this is how it works.
I don't think people realize that. We're going to take another short break, and then, Chino, we're going to come back, and you can rebut everything that Jabran has had to say. Okay, that's Jabran Zahid and Chino Cabalteja,
Yes.
okay, of IFA and SITAR, respectively, here on the SOEST Hour. We're talking about research revelations at UH-Manoa. I'm Jay Fidell. We'll be right back.
Hi, my name is Dr. Rafi. Every week, I'm right here at Thinktech Hawaii, 3 p.m. on Mondays. My show is Boards' Bio Briefings. What do we do here? Well, we watch sperm swim, we see if they catch anybody, we check out the latest biosimilars, the kind that, what was his name, the guy with the bicycle? I guess we forgot his name, but he was taking EPO and other human growth factors. We'll be talking about human growth factors. You want to know where to get some? Maybe I'll tell. Anyway, you can catch me, as I said, every week right here, Monday, 3 p.m., Thinktech Hawaii, Dr. Rafi. You can also find me on Twitter, Bioinfo Medical. Or you can catch me on Facebook, Dr. Rafael Boritzer. I'll be happy to converse with you. Aloha.
We're back. Of course we're back. We've been here the whole time. Where would we go? We have to finish this discussion with Jabran Zahid and Chino Cabalteja of IFA and SITAR, respectively, at UH-Manoa here on the SOEST Hour, which is another school, but a large school, under the rubric of which we do all kinds of scientific discussion. So, okay, Chino, you heard what he said. What part of it do you agree and disagree? He was advocating for his position, right?
Yes. Basically, how we do it in biology is very similar. Whenever I write, I try to make sure that everything that I say is unambiguous.
See, that is a good thing in the whole world.
Yes.
Just to give us, we went to my buddy and me. He's an international relations guy. We went to talk to a group of high school teachers last week, just a week ago, and the conversation, which was a couple hours long, it devolved to one point, and that is how important it is in the world to be able to write what you think. Mm-hmm. And that goes for everybody. You cannot have success unless you can do that. Sorry, go ahead.
Oh no, I totally agree, and it's one of the creative aspects of science that Jabran was talking about, because you need to synthesize your thoughts, but in your head it makes sense. When you start writing it and you start reading it again, it's like, oh, did I really think that, or do I really want to say that? Mm-hmm. And you have to make sure that everything that you say is spot on, because a lot of the work that you do is founded on previous work, and it's kind of a little homage to them to be able to cite their work. And just the other night I was reading a bunch of articles, and I just found it so amazing that the science in my field has come this far, and it's a little I get a little overwhelmed when I read it, because these were like the foundations of our field. Yeah.
That's fabulous. I get overwhelmed just listening. But but if you build on that, sometimes it's a perfect foundation for the next thought, and sometimes it's not. Sometimes it could be slightly wrong, or even more than slightly wrong. So when you write and you cite, do you mention that you agree with somebody you're citing, or maybe you want to correct something he's done, or you want to take it a little further, which he didn't do and should have? Do you ever get into that? I know we want to make friends and have good relationships, but what do you do when you stumble into that kind of problem?
First of all, if something like that arises, I'll reread the paper and see if my views are in agreement or in opposition of theirs. I won't just lambast the research, but I'll give it its due credit and say that, oh, maybe this is an alternative approach to it, or maybe I can add a little bit more to that, and that's how science
You want to be
is.
courteous no matter what.
Yes, sir.
But sometimes we read in the paper about how somebody completely, I mean, recently, I can't give you names, but it happens all the time when somebody is uncovered, you know, somebody is found to be exaggerating something, making up data, misinterpreting data intentionally even. What happens when you find that? Do you just ignore it, or do you call attention to it? If you find it.
I mean, personally, I haven't found anything that big, but usually if I see a paper and it has like a number that's wrong, I'll usually make a correction to that in my papers and say, oh, maybe it was the author intended it to be this figure or this number, or maybe what the author wanted to say was this. I'll give my own interpretation.
So this is part of what we were talking about before with Jabran. This is part of being an excellent researcher. It's looking at the precedent work. It's testing the precedent work, maybe trying to improve it somehow, or even, what did you say, lambaste it? Somehow, to bring the body of science forward. So it's never in a vacuum. It's always in the continuum of advancing science. So if I'm on your committee at CITAR or at UH-Manoa, I should have those things in my mind, right?
Yes. Yeah, definitely. I think it's important for all researchers to just, think critically, but be courteous to each other, because each lab, even if you're studying the same thing, each lab puts in their due diligence, their time doing the work, and that should commandeer some respect.
Okay, let's take a minute and find out exactly what you have been doing, Chino. Can you state, in the law it would be state the case,
Yes.
but in science it's probably state the problem.
Yes.
State the hypotheses. That's what they call it at the science fair. So there must be some parallel between you guys and the science fair.
Yes. So basically, I've been studying for my master's a peptide from a vermivorous cone snail called Conus virgo, found in Australia, and in my peptide chain, I found that it has what we call four cysteines, and it's capable of doing 2 disulfide bonds, and when you find out how many permutations it has in that disulfide bond, it comes out to 3 forms. And what we did in the lab was to figure out whether or not these 3 forms can be generated at random. And we also folded these peptides on the forms that they were intended to be. And
How do you do that?
we basically build it amino acid by amino acid using what we call solid phase peptide synthesis. Take a resin and we attach amino acid after amino acid.
On a microscope?
Microscopic
You
resins.
can't see this
yes.
with your eye.
Yes. So it's actually like little beads, about the size of a grain of sand.
Okay.
And then, we just add it on. It's usually takes us maybe a couple days work. But what happens after that is we cleave it from the resin, and then there we have our unfolded peptide, and we put it in a couple of oxidative buffers, and in hopes to fold it a certain way. And it's kind of analogous to a tie. You have one piece of a long strand of silk, and then you just tie it different ways, and that's basically what we did what I did in my project.
Then what do you do with it?
And we
Test it against possibilities?
Yes, sir.
How do you do that, and what do you test it against?
We'll usually collaborate with pharmacologists from different universities, and we'll send them our peptides that are folded a certain way, and then to see if they have some sort of assay in which there's receptors that they grow on special cells that these ligands bind to and cause some sort of physiological effect.
What's a ligand?
Ligand is something that attaches to another.
Okay. And how do you send it to them? Put it in an envelope, first class? Postal service?
Oh, I think we usually use. Well, I don't think we do just regular post office. We do FedEx, but
FedEx, okay.
we make sure that we seal it properly because there's some regulations that we have to follow in order to ship these bioactive peptides.
You know, I'm reminded, forgive the recollection, but I'm reminded of a medical researcher at Queen's and Jabran who was working with human blood and serum. And he was trying to get diversity data on these, you know, from our diverse population and send it to Big Pharma on the mainland. And he did this for years. I mean, he would put it in a little tube and he would send it to them. And they would pay big bucks, by the way, for that. When you go from, you know, research to industry, there's money involved. Anyway, so and one day he decided that he could do this on a database. So he started examining the blood and examining the serum, you know, in terms of a database with many thousands of fields, and each sample was a record in a large database. It was big data. This is 10 years ago at least. And so I don't have to send them the actual blood or serum. I can send them the database by email. And then they can see all that I know, all my testing about this blood and serum, and they can make their testing hypothetically on the basis of the information. And they paid him the same money for the database that they did for the live blood and serum. I thought that was interesting progress in terms of how you deal with scientific information.
Yes.
And someday you'll be doing that. I hope.
I hope to.
Okay, well let's shift a little bit, okay, and go to Jabran now. Jabran, can you tell us what work you are doing now? What's the work that got you the award, for example? And then you said you published a paper recently. What was that, and was it related?
Yeah, so unlike Chino, I look at the universe at the largest scales, and so my work has largely been trying to understand how structure forms in the universe. And the structure that we see in the universe is traced out by billions and billions of galaxies. And so I study the formation and evolution of these galaxies. Our current view of the universe and its formation and evolution is that we start with the Big Bang, which we don't know much about, although recent results you may have heard of is starting to illuminate some of the earliest physics. But we start with the Big Bang, and very early on, as the universe cools, hydrogen forms, and trace amounts of helium. Everything heavier than that, that means oxygen, lithium, carbon, nitrogen, everything that me and you are made out of, everything that this Earth is made out of, all of that is formed from hydrogen that collapsed into stars, and then that hydrogen was used as the fuel to power the stars, and the way it does that is through fusion. So it takes two hydrogen, puts them together, makes a helium, takes helium, puts that together, makes heavier and heavier elements. So everything that we're made out of was formed in stars, and then these stars explode in supernova explosions, and they seed the galaxy that they're in with these heavy elements, oxygen, nitrogen. And then the next generation of stars form from that gas, and that's how we get something like the Earth. It says, you know, the sun is a second, third generation star, more or less, and you know everything that created the sun and the Earth came from, you know, the stars that came previously. And what I study is I measure how the amount of oxygen that we have in galaxies has evolved as the universe has evolved, to try to understand the chemical evolution of the universe.
Oxygen is important in the development of life, for example, right?
Yes, and also it's the most heavy. It's by mass, 50% of all heavy elements are oxygen, and this has to do with how the fusion process takes place. Oxygen ends up being a very stable element, and so it ends up being, by mass, 50% of all elements other than hydrogen and helium are oxygen, even though it's far down the chain in terms of, you know, it's element number 8, but still.
So it's a good bet to study oxygen, because oxygen is ubiquitous, and
Yes.
it has all kinds of effects on things, and you can track a process forward if you know about the oxygen.
Exactly, and because it's so ubiquitous, it has the strongest signatures that we can measure as well.
Can I find it in peptides?
Most certainly. I'm almost certain you could add.
Oxygen.
I knew there was a common denominator here.
I mean, carbon, nitrogen, oxygen. These are pretty much in anything that's going to be biological and organic.
Okay, so that's your main thrust there.
Yes.
Is that your PhD dissertation? Or?
Yeah. So my work has been focused on making this measurement in more and more distant objects. So the way it works is, the light travels at a finite speed, as that's what special relativity tells us.
It hasn't changed since I went to school, right?
No, so there was a claim that it was it had changed, but it hasn't changed. No, so light
Confirmation, my education.
it was worth something. Yeah, so light travels at a finite speed. So when you look at a distant object, or when you look at an object, you're seeing it as it was in the time it takes light to travel. And when you talk about galaxies and the scale of galaxies, the closest nearby galaxy that's fairly big is the Andromeda Galaxy, and that's 3 million light years away. It takes 3 million years for that light to reach us, and that's the closest galaxy.
The Andromeda Galaxy?
Yes.
Wasn't there a movie by that name?
Yeah, but it was talking about... there you go.
Another common denominator.
Yeah. But so I study galaxies that are much, much further away, and I study nearby galaxies, and in those galaxies I measure oxygen. And so when I study a galaxy that's halfway across the universe, I'm seeing it as it was 8 billion years ago, or 7 billion years ago, and I measure the oxygen in that galaxy, and I compare it to what I see today, and I try to understand and map the evolution of oxygen in the universe as the universe evolves. And
Okay, is this easy? I mean, is this all unfolding before your very eyes, or do you have issues you can't resolve? Do you have questions and problems that you have to apply those creative thought processes against?
I think, this is probably, I'm almost certain it's true for biology, but astronomy is completely driven by technology. So our limitations are always our limitations in technology. And so right now...
You mean like big data? Like huge databases?
Well, that's one aspect of it, but also having telescopes, big telescopes, right? That's crucial. So now, Hawaii is building the 30-meter telescope on Mauna Kea. Yeah.
Because if it isn't, I'm going to be sad.
I think it's happening. I hope so.
Good.
And we're sending up the next generation space telescope that's supposed to follow up Hubble, which is bigger, and it's going to be awesome. You
get up there, huh?
Yeah. Well, this one.
I bet you don't get up there nearly as often, eh?
No.
So I think this is what drives revolutions in science is the technological advancements, and so that's really what's limiting it as well. So I think the next generation of telescopes will open up a whole new frontier for our understanding.
So the interesting part is the technology that you're waiting on. Somebody else is working on that.
Yeah.
You're not working on that.
No.
You're a consumer of that technology.
Exactly. The engineers are working on that.
So do you ever write them a note and say, you ought to do this and that because I could use those tools?
I mean, I don't personally, but the scientists and astronomers in particular are heavily involved in all phases of the development of these technologies, and they have to be because, engineers are concerned about nuts and bolts, astronomers are concerned about science, and they need to both come together to really develop these technologies.
Collaboration.
Collaboration, exactly. And Hawaii is a great example of how this can really work well, right? So
we want to be the collaboration capital of the world.
Yeah.
Right, Talia? Everybody collaborates because it works better that way.
I mean, as far as astronomy goes, it's an international community here in Hawaii, and that's something I think the Institute for Astronomy has really been a huge advocate of, and I think something that we can be very proud of.
Okay, so you're taking a PhD. When are you going to go defend it now?
So the dissertation
Can
defense.
we bring out cameras?
You're welcome to if you like.
No pressure. No pressure.
There's other things I'm worried about, to be honest. No, that's going to be in 4 weeks here. So.
You have to bone up for it?
No, I mean, for me, I've published quite a few papers, so I'm just presenting the results that have already been presented.
Because all the papers are around the dissertation anyway.
Yeah, so they're all related to that subject of measuring and understanding. Actually, that's the key part, is not just making the measurement, but understanding what is the measurement actually telling us.
Explaining. Yeah.
And so the last paper I just wrote was, I think, a big step forward in our understanding of how the universe is evolving chemically.
Okay, let me ask you my last question before I go to Chino as the anchorman here of our discussion. Okay. Why do I care about your work? Why do I care about your work?
That's a great question, and I'd say there's several aspects of it. I think, first of all, we all have a desire to understand our place in the universe. Whether that includes our biological place on this Earth and what role Earth plays in the grand scheme of biology, or how the universe that we live in came to be and how it's changing and evolving. And so I'm directly studying that I'm trying to understand that, and I'm trying to push the envelope. I think, you know, having the picture that we have now has been just remarkable in terms of, you know, it doesn't have a direct impact necessarily on our day-to-day life, but the way our culture is shaped and developed by scientific ideas that give us context for our own lives. I think that's immeasurable, the importance of that. Secondly, I think astronomy has a very special role because I think it's one of the sciences that really attracts people, young people, to math, science, engineering, these STEM fields that are crucial for any society that's going to be successful. Any society in modern day that wants to be successful has to be technologically advanced. And if we have a science that gets people really, really interested and passionate early on, even if they don't go on to do work in that field, I think it still provides a very important tool, very important role in the society in which it's taking place.
It's a culture point.
It's a culture point.
You talk about you know, looking at the heavens. I mean, mankind and womankind, humankind,
Yes.
has been looking at the heavens since day one,
Yes.
and somehow it has always played a role for them.
Yeah, astronomy is venerable in that sense. It's the oldest science. I mean, I think all physics comes out of our attempt to understand the sky and what's happening there. So yeah, I think that makes it something that you should care about. And
I do care.
And you do, I'm sure.
I was there at astronomy open house
Yeah, great.
with Roy Gal.
Yeah, Roy did an excellent job.
Anyway, so Chino, can you top that? First, tell us what your essential hypothesis is, and your writings, and what your master's thesis is about.
Okay. Well, my topic is basically on peptide isomers. As I mentioned earlier, there's a bunch of ways that these peptides can fold because each amino acid interacts with each other in a certain way. And what we're trying to do in our lab is, or what my research is to try and elucidate a little bit more about how that process relates to conopeptides. It's been done with larger peptides, but not with these small bioactive peptides.
Mm-hmm.
And what's interesting is there's a growing field of peptide bioengineering in which chemical techniques are used to basically enhance pharmacological properties of peptides that they want to use for drug leads or pesticide leads. They're incorporating things such as synthetic amino acids. Sometimes they don't even do normal disulfide bonding anymore. They have selenocysteines that do diselenide bonds to basically make the peptide structure more stable. But what I'm interested in is the different folds of the peptide. And we find that some of these peptides with unnatural folds, unlike the ones that the snails usually produce, in nature, snails produce a fold called the globular fold. However, in the lab, we can create folds that are not found in nature. And since we can do this, we can test it against a bunch of assays and see if they have some sort of bioactivity. And when you find bioactivity, it could be a source of drug leads. So now you've effectively expanded the amount of drug leads that you can have from just a normal globular fold or to a slightly different shape. And you can still use these.
This can lead to all kinds of new pharmaceuticals.
Yes, sir.
Do we know what kinds? I mean, is there a wish list out there, or do you just find it as you go down the road?
Well, a big step is to find analgesics, and in terms of drug development, it's to create these new biologics, peptide-based products that are more stable within our system. And the inherent problem with peptides as drug leads is that they can't be taken orally because your body just digests it. So what we're trying to do is basically do some chemical manipulation so that these peptides become more amenable for.
So you have a delivery system.
Yes.
Otherwise, you can make a wonderful therapeutic, but you can't deliver it.
Yes, sir.
Yeah. So, I mean, this is really begging the question, but why do we care? about your science. It sounds like we should care very much on a personal level, because you could cure every disease known to man with this possibility.
Well, we'll try to cure one disease at a time.
This is a smart scientific approach.
Since disease is basically a cascade of a lot of things, and we want to try and see if one we can prevent disease from happening by using these therapeutics, or block one biochemical pathway so that disease does not progress.
This is important work happening right here.
Yeah, yes.
A couple of miles from where we're sitting. That's wonderful. So you're going to finish your thesis pretty soon?
Yes, sir. I'm hoping to finish it by this summer. Well, thesis deadline is July 3rd. So.
Okay, well, you have to go right back.
So hopefully I am married enough to graduate from my program.
Okay, and then what?
And hopefully in the fall I'll be starting another long journey to my PhD.
Which will be where?
Hopefully it's going to be in the University of Pittsburgh in their joint program with Carnegie Mellon in structural biology.
That's a major school in this subject, isn't it?
Yes, sir.
Yeah, good for you.
I'm
Good
very
for you.
fortunate.
Yeah, good for you. And what about you, Jabran? Where are you going to go when you finish your PhD?
So I'll be starting a postdoctoral position at the Center for Astrophysics at the Harvard. It's the Harvard-Smithsonian Center for Astrophysics at Harvard.
Okay, we have a minute left. That's fantastic. We are generating some real talent here, but also losing it. So I hope you guys, when you write your papers, after you take your degrees, you should make some comment about how it all came from Hawaii, and all the seminal thought process and creativity was born right here in Manoa. Yeah, you should give us credit.
Yes, of course.
Anyway, so advice to the kids, okay? Those high school kids who may not have had these thoughts yet, the aha moments. What's your advice to the kids? You can look at that camera.
So, for students who are interested in pursuing a career in astronomy, I think it's important to really focus on mathematics and physics in order to build up your tools in order to be able to then turn to the skies, because we're in a field now that's driven completely by our physical understanding of the world, and you really need to have that solid background. But in general, I would say to the kids, you should do what you want to do. Don't listen to what other people tell you. Please follow your own desires, and if it happens to land you in astronomy, then so be it. If not, then go your own path.
Good advice. Okay, Chino, your turn.
I'd say to those students interested in science, especially astronomy or the biological sciences, to pursue what you want to do. Don't be ashamed to say, oh, I like science. I want to do this. And try to find as much opportunity to be exposed into scientific work, into lab work, so that you'll be a step closer to your dreams.
Yeah, poetry is okay.
Just fine.
You have to write well in poetry, too. That's Jabran Zahid of IFA and Chino Cabalteja
Yes.
of CITAR, the College of Tropical Agriculture and Human Resources, here on the SOEST Hour, which covers everything like this at UH-Manoa. We've had research revelations at UH-Manoa. Great discussion, guys. Thank you very much.
Thank you very much
Thank you,
for
Jay.
having us.
Aloha.